Columbia University, 1930s. The great arched windows of the Chandler Chemical Laboratories are dark voids against the New York night. Inside, long after the salaried professors and their male graduate students have gone home, a solitary light burns. It illuminates a small, fiercely focused corner of the sprawling lab, a space carved out by sheer will. Here, amidst the spectral glow of Bunsen burners and the intricate glass latticework of distillation columns, MaryLCaldwell works. The air is a thick cocktail of chemical reagents, the sharp tang of acetic acid, the faint almond-like scent of benzaldehyde, and the ever-present dusty smell of old books and wooden cabinets. This is her sanctuary and her prison. MaryCaldwell, holder of a Ph.D. in chemistry from this very institution, a recognized expert in the field of amylases, the enzymes that break down starches, is not on the university's payroll. She holds no official salaried position. For her relentless, groundbreaking work, performed in the stolen hours of the night and the quiet moments of the day, Columbia University offers her the grand sum of $0. Her compensation is access, the privilege of using their equipment, their chemicals, their space to push the boundaries of human knowledge. She was a ghost in the machine, a phantom contributor whose intellectual energy was being freely siphoned to fuel the university's prestige. This arrangement was not an anomaly. It was the system functioning as designed. The Great Depression had choked the academic job market, and for women, the prospects were nearly non-existent. A woman with a doctorate was an inconvenient reality, an exception to a rule that saw the lab as a male domain. The prevailing attitude, documented in countless university correspondences from the era, was that women in science were, at best, a hobbyist class. They were presumed to have husbands or fathers to support them, making a salary a secondary concern to the enrichment of intellectual pursuit. For Caldwell, a woman who would never marry and who dedicated her entire life to her work, this assumption was not just insulting, it was a fundamental erasure of her professional identity. She was permitted to conduct research, to mentor students, many of whom would go on to have illustrious, well-paid careers based on her teachings, but she was denied the basic dignity of a contract, a salary, a title that matched her expertise. She was a research associate in name only, a title that carried the weight of expectation without the support of remuneration. Her existence was a paradox, essential to the laboratory's function, yet invisible on its official ledger. Every successful crystallization, every meticulously recorded data point was a gift she was expected to give freely. Her focus during these years was on enzymes, the biological catalysts that drive the chemical reactions of life. Specifically, she was a world authority on amylases, enzymes found in saliva and the pancreas. Her work was painstaking, a masterclass in patience. It involved purifying these delicate proteins from sources like pancreatic tissue, a messy, smelly, and incredibly precise process. She would grind, centrifuge, filter, and precipitate, step to agonizing step, to isolate a single pure substance from a complex biological soup. This expertise in purification, in separating the essential from the extraneous, was more than just a technical skill. It was the foundation upon which the golden age of nutritional science would be built. At the time, the scientific community was in a frantic race to identify and isolate a group of mysterious compounds they called vitamins. They knew these substances were essential for preventing horrific diseases like scurvy, pellagra, and beriberi, but their chemical nature was largely a mystery. The so-called vitamin B was a particular puzzle, believed to be a single entity responsible for a wide range of biological functions. But researchers were beginning to suspect it was not one substance, but a complex of many. To solve this puzzle, they needed chemists with an almost supernatural talent for purification. They needed someone exactly like MaryCaldwell. And so, the world-renowned nutrition researchers at Columbia, like Professor Henry C. Sherman, turned to the ghost in their own laboratory. They needed her mind, her hands, her unparalleled skills. And they knew they could get them for free. The question that hung in the silent, chemical-scented air of the lab each night was not whether she could make a discovery, but what would happen to that discovery once it was made. Who truly owned the insights forged in the crucible of this uncompensated, unrecognized, and utterly brilliant labor? Following those grueling, unpaid hours, a period of thankless intellectual devotion that would stretch for nearly 2 decades, Caldwell's tenacity began to cleave a path through the scientific unknown. The puzzle of the vitamin B complex was yielding to the combined efforts of labs across the world, and her unique expertise in purification became the critical key for the Columbia team. By the late 1930s, the race was on to isolate a specific factor within the complex responsible for curing a type of severe dermatitis, or skin inflammation, observed in rats. This condition, known as rat acrodynia, became the biological assay, the living test tube for the discovery. The process was a brutal, iterative form of alchemy. Feed a rat a deficient diet until it develops the painful skin lesions, then feed it a purified extract from a source like yeast or rice bran. If the lesions healed, the extract contained the anti-dermatitis factor. The challenge was purifying that factor down to a single crystalline molecule. This is where Caldwell's genius shone. Her methods, honed over years of isolating delicate enzymes, were perfectly suited for this task. She employed techniques of fractional crystallization and selective adsorption, meticulously separating compounds based on minute differences in their chemical properties. Her lab notebooks from this period, held in university archives, are a testament to this rigor. Page after page of precise measurements, failed attempts, and methodical adjustments. This foundational work culminated in a landmark 1938 paper published in the German with Paul Jurgi and other collaborators. While Jurgi is often credited with naming the substance pyridoxine and others with its synthesis, a close reading of the experimental methodology in these early papers reveals Caldwell's intellectual DNA. The paper details the painstaking process of isolating a crystalline substance with potent vitamin B6 activity. While her male counterparts received the lion's share of funding and recognition, it was Caldwell's mastery of biochemical purification that made the isolation possible. Her name is on the paper, but the academic hierarchy of the time tells a different story. As a woman, and one without a salaried position, her role was systematically downplayed. She was cast as the skilled technician, the good pair of hands, while the men were lauded as the intellectual architects. The systemic resistance wasn't just about a paycheck. It was about the deliberate curation of scientific legacy. It was a story told in the order of authors on a publication, in the invitations to speak at conferences, in the awarding of grants that listed a man as the principal investigator, even when the core ideas and labor were hers. This injustice is starkly illustrated by her official status at Columbia. It was not until 1948, after more than 25 years of continuous high-impact research and teaching, that the university finally granted her a salaried position as an assistant professor. A year later, she was promoted to associate professor, but she would never, ever be granted the title of full professor, the highest academic rank, a rank bestowed upon countless men she had trained and whose work was often far less significant than her own. The institutional gatekeepers had allowed her to contribute, but they had built a ceiling of glass just above her head, ensuring she would never be seen as an equal. The discovery of vitamin B6 was a collaborative international effort, with researchers like Richard Kuhn and Albert St. Georgi also making crucial contributions. But collaboration requires that all contributors are seen and valued. The system at Columbia and at universities across the country was designed to render the contributions of women like Caldwell functionally invisible. It absorbed her labor, extracted her intellectual property, and then wrote her out of the heroic narrative of discovery. The molecule was now in the world's hands, a tangible crystalline powder that held the power to cure disease. But the architect of its unearthing remained in the shadows. What happens to the truth of scientific progress when the official record is written by those who hold the power, not by those who do the work? That shadow of anonymity would stretch for decades, even as the molecule she painstakingly helped isolate began to fundamentally reshape human health. The isolation of pyridoxine was not an end, but a beginning. It transformed vitamin B6 from a mysterious nutritional factor into a tangible chemical that could be studied, synthesized, and deployed. Building on the foundational work of Caldwell and her contemporaries, the full, staggering significance of B6 became undeniable. It wasn't just a nutrient that prevented skin conditions in rats, it was a master key in the engine of human biochemistry. Her foundational work on enzymes was prophetic because we now know that the active form of vitamin B6, pyridoxal 5-phosphate, is a coenzyme, a helper molecule in over 140 different essential enzymatic reactions. Think of it as the universal adapter for the cell's machinery. Without it, the entire factory of life grinds to a halt. For instance, B6 is absolutely critical for amino acid metabolism. It allows our bodies to convert the building blocks of protein from one form to another, a process essential for building muscle, creating hormones, and repairing tissue. Every time you eat a protein-rich meal, the ghost of MaryCaldwell's work is present in the silent, efficient chemistry taking place in your cells. Its impact extends deep into our nervous system. Vitamin B6 is indispensable for the synthesis of key neurotransmitters, the chemical messengers that regulate our mood, our sleep, and our thoughts. It is a necessary component in the creation of serotonin, the feel-good neurotransmitter, dopamine, which governs motivation and reward, and GABA, which has a calming effect on the brain. Deficiencies in B6 are now linked to depression, anxiety, cognitive decline, and certain forms of epilepsy. The modern fields of nutritional psychiatry and neurology, which explore the profound connection between what we eat and how we feel, stand directly on the shoulders of this discovery. Furthermore, her work laid the path for understanding B6's role in red blood cell formation. It is a crucial component in the synthesis of heme, the iron-containing part of hemoglobin that carries oxygen in our blood. A lack of B6 can lead to a specific type of microcytic anemia, leaving a person perpetually fatigued and weak. From preventing anemia to supporting brain health, the ripple effect of her discovery touches nearly every aspect of human well-being. Today, pyridoxine is on the World Health Organization's list of essential medicines. It is a standard component of prenatal vitamins, crucial for fetal brain development. It is used to treat nausea during pregnancy, to counteract side effects of certain medications, and is studied for its potential role in immune function and cardiovascular health. Yet, despite this monumental legacy, Mary.Caldwell's personal story ended not with a Nobel Prize or a prestigious chaired professorship, but with a quiet retirement in 1959. She left Columbia as an associate professor, the glass ceiling above her never having been broken. The system had taken what it needed and had given back the bare minimum. Her name is a footnote in textbooks, if it appears at all. Her story is a stark reminder that scientific progress is not a pure meritocratic endeavor. It is a human one, shaped by prejudice, power dynamics, and the biases of its time. It forces us to look at the crisp, clean molecular diagrams in our textbooks and ask a difficult question. Whose labor is hidden behind this elegant structure? Whose late nights, whose unpaid work, whose silenced voice made this knowledge possible? Her story forces us to confront a profound question. How many lives have been improved by the breakthroughs of brilliant minds whose names were deliberately systematically erased from the annals of science? Remembering is not just about correcting the past. It's about building a more just and effective system of discovery for the future.